Hey, hello everyone. Welcome to Chapter 6 video. Today I'm going to be covering the first half of this chapter, and then the second half we'll do in a second video. So guys, this chapter is going to cover cell division. If you remember from chapter number 4, we talked about the cell, we talked about all the organelles that are inside the cell. So today we're going to see how does one cell divide itself into two cells. And for that, what steps does it need to go through? So you're going to talk about the cell cycle, okay? So the cell divides for a couple of reasons. There are two important reasons. One is that the organism wants to reproduce. You want to go from one organism to two organisms, so that's reproduction. The second reason could be there's a already existing body which wants to grow. So let's talk about, let's say, an infant growing to an adult. We started with fewer numbers of cells, and then as our body becomes bigger and bigger, we are putting in more number of cells. So growth needs more cell. Also imagine, guys, when when the baby is born, that came from one cell, which is called zygote. So zygote divided multiple times, and then that in result created billions of cells, and that's what makes up a small baby, right? So for growth we need cell division, and also for repair of cells. Once you have the whole organism, we need to repair. Let's say you got we know that our hairs grow, that needs cell division, or our skin cells needs to be replaced. That's when we get cell repair, so that also requires cell division, and many other things that require cell division, okay? So the first thing we're going to talk about is a cell division that takes place in usually prokaryotes, for example bacteria, and then also in some single cell eukaryotes, okay? Now the very first type of cell division, guys, is the very basic one. This is called binary fission, and this is also considered an asexual reproduction. So guys, asexual reproduction means that it requires just one cell. There you see that there's only one parent. So it requires one parent only. That's your asexual reproduction. In asexual reproduction, what happens basically is you have the cell, you got this DNA. One cool thing to notice, guys, here is that it is a circular DNA. Since we're talking about bacteria, guys, has a circular DNA, whereas we eukaryotes or humans, we have linear DNA, okay? So that's just one thing to notice. What happens next? So we want to take the cell, the parent cell, and we want to parent cell, and we want to divide this into these two daughter cells. So that's reproduction. Now, why is this asexual again? Because this only involves one parent, in contrast to sexual reproduction in most of the plants and in most of the animals. In eukaryotes, we know that there are two parents involved, and that's considered sexual reproduction, okay? So we got sexual reproduction, which requires two parents, so mom and dad, okay? Whereas in asexual reproduction, it's just one parent. Okay, so now for cell division, what do we need, guys? We need to first double the content. So we got this DNA inside. We first need to make two copies of that DNA, and then the cell is going to pinch off. You can see the cell wall, cell membrane pinches in, and then this cell divides into two different cells. Now we got these two cells. And the thing to notice, guys, that in sexual reproduction, the daughter cells, guys, that are produced, they are genetically identical. What does that mean? This DNA material is exact same as this DNA material, which is exact same as this DNA material. So the siblings, so the two daughter cells that are made, are similar, or not similar, they're exact same to each other, and then also they're exact same as the parent. Okay, so the two daughter cells from bacteria are going to look the same as the parent cell. Okay, all right. So that was your asexual reproduction. There are other methods as well for asexual reproduction. Like this is called binary fission. There's also budding, or fragmentation, but we will not talk about those ones. Okay. All right, now let's talk about the sexual reproduction. And now this happens in eukaryotes. So when our cells, sorry about that. So when our cells need to divide, they need to go through what we call the sexual reproduction. Now, our focus right now is not just going to be on reproduction, but also growth and repair, as I mentioned before. So guys, we have mitosis and meiosis that takes place in our body. Most of the cell growth, or when the organism is produced, only mitosis is what takes place. And the other one, when our body has to produce gametes like sperm and egg, that's when meiosis takes place, okay? But we're going to talk about mitosis and meiosis in our next video. So before mitosis and meiosis, guys, there has to be some other stuff that has to happen, okay? And that is what happens before cell division, okay? So our cell cycle is basically divided into these two different stages. We got this interphase, and then we got this cell division. So guys, interphase is the time when, so most of the time the cell spends in its life is the interphase, okay? So at the end, the cell has to divide. Let's talk about RBC, so red blood cell, which only survive for 3 months. So at the later stage, that's when they will, or your bone marrow cells are gonna produce the red blood cells, okay? But before that, the cells will just stay in the interphase, okay? All right. So so this is when all the cells are just doing their own job, like whatever they're supposed to do, and then at the end they will get ready for cell division. Now, cell division is the last stage of the cell cycle, and that's when your DNA first needs to be duplicated, and then it's divided into 2 cells. Okay, so basically one cell breaks into 2 cells, becomes 2 cells. Okay, so if we see that here, let's take the next slide. So our interphase, where the cell is going to spend most of the time in its life, consists of these 3 different phases, okay? We got the G1 phase, we got the S phase, we got the G2 phase. So G1 stands for gap 1, and then your G2 stands for gap 2, like here. And then the S phase stands for synthesis. So this is when your DNA needs to be synthesized or replicated. So there's already 1 copy of DNA. It's going to be replicated into another one, okay? So this is the first phase after the cell divides. So this is a newborn cell, and it goes through the G1 phase. This is the longest phase. So it's now getting ready for the DNA to replicate. So it's the phase when all the material will be collected, all the raw material that is required for synthesis of DNA and other stuff in the cell, all that will be collected, okay? So if we see in the cycle here, you should become familiar with this cycle. You're going to see it on your quiz and also on your exam, and you will be asked to fill out like what happens in each stage. So you should be very familiar with this figure, okay? So the very first, as you can see, the G1 phase, this is our G1 phase right here. This is when your cell has grown and it's ready to duplicate. So the cell is pretty big at this time, and then you can see this is the DNA thing right here. This DNA is called chromatin. We'll talk more about that in our next slide. Then guys, after G1 phase, cell goes into the S phase, which was the synthetic phase, and the DNA replication is what takes place here, okay? So you should remember S with synthesis, so that's where DNA synthesis is taking place. The next phase is the G2 phase. G2 phase is when it's made sure that when you went through the S phase, the DNA replicated properly and all the raw material is required so that we are ready to go for the cell division, okay? So this phase right here is what the cell division is, okay? So cell division includes mitosis and cytokinesis. In mitosis, guys, our DNA will be separated on the 2 sides so that our DNA material can be given to 2 separate daughter cells. And then cytokinesis is when the actual cytoplasm division takes place, and you can see the cell actually, the membrane actually pinches towards inside and actually breaks into two different cells. So that process is cytokinesis. Okay, so you should be familiar with each of these phases. Okay, let's see. So this is, again, we're talking about the interface, and we're going to talk about the S phase. What is important here, guys, is to realize what do we mean by all these different structures that we are seeing here. It's very, very important for understanding, not just for this chapter, but for the coming chapters as well. Okay, so first of all, you got your DNA. This is your DNA sitting inside the cell, and they're showing you that this is during the gap phase. Okay, so and then you go to the S phase, the synthetic phase. So what is this showing here is the DNA, which is just sitting like a thread-like structure. And guys, this thing, we call this guy a chromosome. Okay, now chromosome, what does it mean? So chrome actually means color, and zome means body. So guys, when first people stained the cells with some color and they saw it under a microscope, they actually saw very bright, colorful bodies, and then they started calling that chromosome. Okay, at that point, of course, they didn't have much idea about what they actually do. Now, we got this chromosome, so that by itself, and then what happens in the next stage, guys? Our DNA replicates into two. So what happens is there's a protein in our cells which starts making copy of this DNA. Okay, so it's going to sit right here, and it's going to start making copy of this. So it's going to start making copy for this whole chromosome, and it results in giving you two separate DNAs. Okay, now what happens? Now when you made the copy of the whole thing, in the very center, guys, is actually bound together the two. The copy of the DNA that you made from the existing DNA. So one was, of course, the parent DNA, so the actual parent DNA, and then you made a copy of that. Both of those strands are actually held together by this structure called centromere. Okay, so this thing is also called chromosome. So this is also chromosome. This is also chromosome, but this chromosome has double the DNA, has double the content. Okay, now. Let's also go over this terminology of what do we call these two guys. These two guys are called sister chromatids. Okay, see, they were just born from each other. I mean, this DNA was made from this guy, so they're called sister chromatid. Okay, so when you see this thread-like structure, we call that chromatid. Now, one whole thing together is called chromosome. A chromosome can either have two chromatids, and you can call them sister chromatid, or you can have a chromosome which is just one strand of DNA. Okay, I mean one double-stranded DNA, of course. Now, what happens after synthetic phase? So guys, this is when replication. So this is called replication. Okay, when your DNA is copied to make sister chromatids. Okay, now when you go to a later stage, in our later stage, guys, this is the when we want to go into mitosis, we want our DNA to be very tightly packed. Okay, so right here, the DNA is loosely packed. Okay, so there's loose packing of DNA. Okay, but here it's tightly packed. So when we enter mitosis, in the very beginning stages of mitosis, in order to keep things less messy, the DNA is, with the help of proteins, and we're going to study that later, they're tightly packed so that they can be moved around inside the cell more easily. Okay, because if you try to move these guys when the DNA is all open, it's going to be hard to move this guy, but when it's packed, it's easier to move. Okay, all right. Then guys, there's also this stage called G0 phase, or G0 phase, if you want to call that. So guys, it happens right after, during the G1 phase. So this is when the cell was divided, a new cell was made. That new cell that was made can actually go into this G0 phase, so it actually exits the interphase or the cell division. So it does not want to divide, and it just wants to stay like that. And actually can spend all of its life in the G0 phase. So imagine the brain cells after the cells are made, they usually don't go for more cell division. They just if you got, it said that up to 5 or 6 years of age, you made as many neurons that you could make, and after that the cells permanently stay there as it is, so they don't divide anymore. Although now it's being said that they do repair themselves, or they can even regrow other cells. Especially that it's a hot topic to study, especially with the people with brain damage and stuff like that. So many adult cells which have specific functions, like we just mentioned, neurons in the brain, so the brain cells, they can go into this G0 phase and stay like that for the rest of the life, and they don't divide anymore. Okay. All right. So what happened? I mean, if you want to think about this, guys, how does a cell know that it's ready to go into the S phase from G1 phase, or it's ready to go from G2 phase, or to go into mitosis, or it's ready to go from S phase to the G2 phase? There are checkpoints that are there in the cell. So there are some proteins which will go around and make sure that things that are happening with the cell, with the DNA and everything else is going well. Otherwise, it's going to stop the cell cycle right there. Okay, so these are regulatory proteins, and they're very, very important. Okay, otherwise we can mess up our cells homogenesis. Okay, all right. So if we talk about the G1 checkpoint, so they will stop the cell cycle if the cell is too small, so it's not ready for cell division, or the nutrient supply is inadequate, so there's not enough raw material there to make more stuff inside the cell, or the cell DNA is damaged. Any of these things could happen, and the cell will just freeze at G1. It won't go to S phase. Okay. Now, even after it goes to the S phase, we have this G2 checkpoint. So this stops again if the cell is too small, if there's no nutrient, not enough nutrient, or the cell is damaged, or if the chromosome that duplicated in the S phase is incomplete. So duplication is not done yet. So replication is not done yet. So there will be a checkpoint right here, and those proteins are not going to allow the cell to move from S phase to G2 phase, and that's going to check the cell cycle. It's going to stop the cell cycle right there. Okay. So again, you can see that in this figure, we got some G1 checkpoint, G2 checkpoint. So there you can think of this as there are some hormones that are acting like a brake, and then they prevent the cell division, or you can think of some hormones and growth factors which actually act like a gas pedal in a car, and they push the cell towards the cell division. So there are some factors which will make you pass through from one cell cycle, from one stage to the other stage, and let the cell cycle go through, and then have cell division. And then there are some hormones which are actually just going to do the opposite and stop the cell division, and that's why we call them checkpoints. So they regulate the cell division. Okay. Now, when these go wrong, things like tumor and cancer can happen. Okay. So what is tumor basically? Tumor is when the cells mass created very rapidly, and they go through cell division more often, so they don't stop. They just keep growing, and that's why they create so much more mass. So one cell is going to divide into 2 cells, 2 is going to divide into 4 cells, and this doesn't stop, and that's why you create a big tissue which is made up of lots of cells which are not supposed to be there. Okay, so now the tumor cells, just to understand, they can be malignant or they can be benign. Malignant is, guys, when the cells actually detach themselves from where they started growing, and they can go through the different parts of the body, and then they can invade other tissues, and that's called metastasis. And then they can even promote angiogenesis. Now, what does angiogenesis mean? Angio actually means blood vessels, and genesis means to produce more blood vessels. So these tumor cells, they're going to cause angiogenesis so that they can get more food supply, and that's why they keep growing more and more. On the other hand, there can be some tumors which are benign. Now, benign are the less severe ones. So they're the one, they remain confined to one site. Let's say somebody has a tumor, a benign tumor in one organ. It's not going to go to another organ and infect that part as well. So it's just going to stay at one place. Okay. And on the other hand, the malignant were the ones which actually move around, and they can go from one tissue and then invade another tissue and cause more tumors over there. Okay. Right. So guys, I'm going to stop right here for this video. And then for the second video, guys, you're going to learn more about cell division, and then we'll actually talk about mitosis and meiosis, which are a very important topic. Okay. See you guys in the next video.